Hello, my name's Adam Smith, and it's my very great pleasure to welcome you to the Nobel Prize Inspiration Initiative. The initiative is a partnership between Nobel Prize Outreach and 3M, designed to take Nobel laureates to meet the next generation of scientists. Today we have 3 Nobel laureates for you in the fields of physiology or medicine and chemistry. This session will be divided into 2 halves. For the first 30 minutes, I'm going to interview the laureates to get them to introduce themselves a bit, and then the next half will be dedicated to your questions. You can start submitting those questions now using the dialog box on the right of your screen, and we encourage you not to be shy and to send in anything you want to ask. The more questions, the better. I would like to thank very much the Austin Independent School District for organizing this and for letting us meet you all. And now let's meet the laureates, Bob Grubes, Andy Fire, and Liz Blackburn. I think we'll begin by having you talk about your work a little, and let's do it in ascending date order of Nobel Prize. So, Bob Grubes, you were awarded the Chemistry Prize in 2005, so you have the honor of going first. You were awarded the prize for the development of a new method in organic synthesis, metathesis. Could you tell us very briefly what it was about?
Yes, thanks. Yeah, and so I'm an organic chemist, and organic chemists like to make molecules which involve making and breaking bonds. And metathesis provides a really new way of doing that. That allows you to make molecules that range from pharmaceuticals to plastics and polymers. And so we developed this catalyst. I've studied it my whole career for 50 years, and in the early 90s, we made our first catalyst, and we've been making them better and better since then. But essentially, based on this new way of constructing bonds, it's resulted in, for example, the first hepatitis C drug had a component that was made using chemistry we had developed. There's 40 miles of pipe in the bottom of the Gulf that is coated with a plastic that's made using the catalyst, and the largest refinery in the world that uses seed oil is going in Indonesia using the catalyst. And so basically, finding a fundamental way of putting carbon-carbon bonds, breaking them and putting them back together again in new ways opened up lots of possibilities.
Thank you very much indeed.
Yeah.
Okay, next to Andy Fire, who was awarded the Medicine Prize in 2006 for finding a new way of silencing genes, RNA interference. Andy, tell us about it.
So I was one of many people who were working on situations when genes would suddenly turn themselves off for various reasons. And often we didn't understand why. And sometimes we knew that we had intervened in the system by putting in a little bit of DNA or a little bit of RNA. And what I and many others in the field did was to try and figure out what kinds of things triggered that event. And my colleague Craig Mello and I and some of our other colleagues at that time, University of Massachusetts and Carnegie Institution, actually figured out that if you had both strands of RNA for some reason, this gene silencing event was very, very efficient. And we named that RNAi after Craig did an online poll, actually. And that's essentially one of the ways that we have now of sort of doing the opposite of what Bob was talking about. We break bonds in a specific way. We break bonds in an RNA that we can target using the sequence of that RNA. And if you do that with just the right sequence and just the right delivery method, you can actually make change in a specific way in biological systems, which is what RNAi has been used for.
Thank you very much indeed. Beautifully succinct. Thank you. And Liz Blackburn, you were awarded the Medicine Prize in 2009 for your work on telomeres, the caps at the ends of chromosomes, and discovering this enzyme telomerase. Please tell us about that.
Well, there was a mystery in DNA, which was that nobody knew how the very ends of DNA were replicated. And there was another mystery. Nobody knew what the DNA at the ends of chromosomes was. And so with a fortuitous system that allowed me to analyze the molecular nature of chromosome ends, actually by using very small chromosomes in a single-celled organism, I was able to find that it was a very unusual kind of DNA sequence, and particularly strange was that the number of nucleotides at the ends of DNA molecules, the ends of chromosomes, were changing, and so, long story short, we found that part of that change involves adding DNA to the ends of chromosomes by a then unknown enzyme that we later called telomerase. So I see a bit of a theme here about bonds. This is an enzyme that makes bonds. It adds DNA to the ends of the long DNA molecules of chromosomes. And how that all played out was the work of a great many people in terms of what that meant in terms of how our chromosomes are maintained during our long lives as humans. And essentially, the DNA replication process makes chromosomes more likely to shorten over time than you would hope. And it does happen, and telomerase adds back DNA. And we now know that we have to have intact caps at the ends of chromosomes with enough of this particular telomeric DNA in order to keep the chromosomes stable. And all sorts of things happen to our health and our disease processes if this is not a process kept in the right balance. So we have to make sure our chromosome caps are good and stable.
Thank you all very much indeed. When I was thinking about getting you all to describe your work, I was thinking, how am I going to bring you all together with these disparate pieces of work? And you've already done it you've done the job for me by bringing up this bonding idea.
We bonded over this,
We bonded, okay.
Yes, right. It's all chemistry.
Beautiful. Even online it works, even virtually. Isn't that nice? Yep. It's the power of science to unite. So people will get the chance over the next 2 days to ask you lots of questions about these things, which is lovely. But the one thing I was thinking that probably does bring you all together is your motivation for doing this work in the first place. The results of your work, the applications are many and various, but why you actually started to pursue these questions. Was it with application in mind, or was it something else that drove you?
I mean, for me, I grew up in a rural environment and went to college to be an ag major. And fortunately, got to learn a little bit about chemistry. And then I discovered organic chemistry and was just fascinated by the intellectual component of it, which is this construct that's been put together that allows us to understand many different reactions and make predictions. But also there was a manual aspect of it too, which was going in the lab and making things and making new stuff. So the original thing for me was this combination of which fit my background, which was understanding how one thinks about molecules, how you make predictions, and then also how you go in the lab and then translate that into something. And the other thing too, which I laugh about sometimes, I was a really bad student. It was the first thing I found I was really good at.
Well, you were really good at it.
Yeah, okay.
Andy.
I mean, it's not totally that I just like to break things, but it can be fun if you are doing it in a guided way that's interesting. So, but I think looking at pattern and specificity was always something that was beautiful to me in biology, and being able to change things in a directed way was always of interest as an intellectual exercise and something to actually a tool to understand biology. At the same time, it was clear to me that the most transformative science came from experiments that were a little bit off the wall, a little bit unusual. And so I've always tried to do some of those experiments, and many of them haven't worked, and many of them they haven't worked because I did the wrong experiment or wasn't doing the right thing. But I certainly enjoy being able, when I can, to go into the lab or onto a computer and try something, ask a question, and get an answer that I don't expect. And that's what I enjoy about biology when it tells me I'm wrong.
When did you get the confidence to do that, to play and not mind being wrong?
it's more that I was fortunate to work in environments where that was supported as even starting with my undergraduate work and at a department where Liz was a faculty member actually at the time, and then moving forward as a graduate student and a postdoc in communities where that was a respected part of life. I hope we still respect that in science. Sometimes it's hard to justify giving resources to someone who not only might be wrong but tells you, I don't know if this is right. I'd like to try it. Can I have the resources to, like, my time for the next 2 weeks to try this? That's a valuable position to be in, and I respect the people and institutions that try and make that kind of environment happen.
Thank you. Liz, please.
At the time I started the work on chromosome ends, really very little was known about DNA sequences, and certainly nothing was known about what was at the ends of chromosomes. So it was really a question of curiosity. I just realized that there were some chemical and enzymatic tools available that had just been developed as techniques for the very earliest ways of sequencing DNA, laughably crude and simple as we look at DNA sequencing now. But it was now it was possible to ask a question that nobody had had any ability to ask before. What is at the ends of DNA molecules that are naturally occurring inside cells and not bacterial cells and not viruses? And so I just wanted to know what was there. And then it just kept going because what was there kept on breaking rules. And I love the mystery of it, and I love the excitement that you didn't know what you were going to find. And I think that's one of the best things, as Andy is saying, about basic science, that the real joy is when you find things that you didn't expect to find. And so your curiosity and imagination get greatly piqued by these novel kinds of things that are showing up and are telling you this is what life is doing, this is what biology is doing. Let's try and find out why. Certainly it was with no thought of any particular application in human health or disease processes. It was just, let's try and find out. So that setting where the National Institutes of Health just said, go for the science, try and find out what you can, was wonderful and really enables science to happen in ways that you can never predict.
Thank you. Bob, please.
Yeah, I mean, just to comment on that, when I tell my students, if you run a reaction and you get what you expect, you haven't learned anything. But if you get the unexpected, then you really learn stuff.
I'm interested in this confidence thing because, in a way, I mean, Nobel laureates often point out that everybody starts out curious, and they're all driven by curiosity when it comes to it. But curiosity can sometimes be a little bit ironed out by school, studying for exams, and needing to know the right thing to get the right grade and all of that. Do you find that when you have students coming to your labs that they can sort of relearn curiosity, or are the good ones who are going to do well already so curious that it's just obvious that they've got this insatiable desire to know?
Well, I could comment on that. I don't think they all come in with a one-size-fits-all, I'm curious. I think that some come in with very different motivations, and having an environment of training where you can say, it's important that you explore and you develop your own ideas, and a lot of science will be failure. I think one of my most revealing moments was I looked up in my notebooks, because I was asked when I got the Nobel Prize, would I look up in the notebooks, and I go through my notebooks and I find that 90% of the things probably I tried didn't work. And so somehow our memories are enabling us to be very resilient to what I can call, what seemed like failures at the time, which is that you sort of just keep going with the ones that do lead you down productive paths as much as you can. But a lot of ability to, quote, fail, I think, is very important. Otherwise, if you're not failing, you're not pushing boundaries. So I think we need to understand that basic science looks inefficient from the outside, but it's really important to be able to just push the boundaries. And sometimes a failure later on, you say, aha, now that feeds into this question or this kind of experimental approach in ways that you didn't expect. So I think this confidence issue is, you have to have people understand that it's really okay to keep pushing, so long as you're thinking well and thinking rigorously and doing it as well as you can. That kind of failure, I think, is what is part and parcel of pushing boundaries.
Thank you.
I mean, I feel like I came up in a good time when there was sufficient funding so you could fail. I'm hoping now with the pressures on funding and the cutting of funding that one has to be much more certain and there's much more tolerance of failure. I mean, much less tolerance of failure. Yeah.
That's interesting. Andy was also hinting on a possible change in the environment now. Let's turn to the environment. I mean, another thing that unites you is that you've all chosen to live in California. I was reading an obituary for the statesman George Shultz the other day, and apparently he was at Stanford, and apparently when ex-president of Russia Mikhail Gorbachev visited him, he said, "Ah, George, I see you have chosen to live in paradise." So here you all are in California, living in paradise. But how important is it to find yourself in a place that is really sort of well funded and everything, has all the advantages, or can you do great science without being in that sort of place?
My contributions in RNAi were I was born in California. I love California. It is not paradise. Anybody who has dealt with our fires in the last few years, our politics, anything is going to know this. One can do science in a lot of different environments. The people are the most important component of it, and that's the environment. That's really the environment as it's defined. Money can be the end of things. If there's too much money and too much responsibility to account for it, it actually becomes a detriment to doing good science. And so the groups can get too large, the problems can get too diluted. And so there really is an advantage in working in a small environment where what is the best thing for me is to be able to come and think about a problem, tinker with it. You do need tools for it. The tools do, in some cases, cost money. You need colleagues to bounce ideas off who are going to be accepting of really crazy ideas, but also be critical and try and improve them. And that can come in a lot of different places in a lot of different forms. And that's why you see people from different environments. California is a place where a lot of scientists have converged, but other places as well. And you can see a lot of really great science being done throughout the world in different environments. So I would not say California has anything unique. Everybody come visit. Before you decide to live here, spend a fall here with the fires happening.
It's really people. And, when I think about the practice of science, if you will, over the years, the thing that stands out is just the wonderfulness of talking about science with colleagues and bouncing ideas off and just having that process of human minds coming up with, more and more interesting kinds of things that honestly by yourself you wouldn't have dared necessarily to do, or you wouldn't have thought it through properly. And so, some of the really good science is done because you have to think in elegant ways because you don't have infinite resources. And so you have to think much more smart about it. And that can make you into a good scientist. But it's certainly not unique to California. And over the years, I remember being struck by the fact that, I'd go and give a research talk, a lecture at various places around the country. You'd be invited to talk, and then you'd spend a day or two talking with scientists there. And first of all, I was thinking, why am I getting on a plane and going to this place and talking? And oh gosh, I could be back at home in the lab or with my family or something. And then you'd realize every place you went, no matter how sort of seemingly obscure, you would have had very likely at least one totally unexpected conversation about science with somebody you'd never met before, whose research you never knew, and you realized there were just tremendously smart people everywhere. And so I like to sort of have this image of really good science just bubbles up everywhere if you let it bubble up. And so I wouldn't be at all restricted by saying, oh, it's California. There are places everywhere, no matter how small and obscure, where it's the minds and the thinking of people that really just shine.
Bob,
Yeah.
please.
Yeah. I mean, I moved to Caltech for the students and the people, and it could have been any place. I'm certainly happy it's in California, so I can go walk and enjoy the outdoors and fish and do the other things. But it's really down deep. It's the people. And so recently, I had a student who had two job offers. One was a very good salary, lots of startups, and the other one, it was at a phenomenal place that had really, really good students. And my advice, which I think he takes, is that you go for the students. The money runs out soon. It's the students, and it's the quality of students you work with. And having the ability to sit down in an office and starting talking about one problem and ending up on another one, and people are willing to take new chances. I've changed. I've gone into a lot of different fields, and it's because the students are willing to do that. And sometimes I tell them their hardest job is going to be teach me.
Now we're on the subject of talking and sharing your science. I suppose as a young person, it's often a little bit difficult to know how much you should tell people about what you're doing and how much you should keep to yourself in case somebody takes your idea. What's your advice? Should you just always talk? Andy?
I mean, I had a wonderful graduate advisor who used to say that, it was much worse to be ignored than to be scooped. There are times when an idea is more ready to talk about in one environment than another environment, but there's rarely an idea that's so unique and so perfect that it wouldn't benefit from feedback from people. So it's usually more a matter of trying to find the people that can give the best input on something rather than to try and hide it from somebody. You're trying to get it to the most useful person to be able to give you feedback on it over time.
I heard it once said that in business you try and kill your competitor, and in research you actually want your competitors to thrive, because that's making even better science happen. And I think there was some understanding about this in the computation software industries, where when things were being developed very early, they were open source because people realized things would move faster and better. And so I've always felt, yes, you should share your ideas. You might get scooped, but as I would tell my students, who more or less believe me, I'd say, look, what will be remembered is the good paper. That's the paper that will prevail, not necessarily the one that came out, a few months earlier or something. And so I've always felt it's always been so much more rewarding. I mean, I just talk to people. Maybe I'm just a blabbermouth and I can't stop, but but I'm excited. I want to talk with people, and it pays because you get such interesting input. And biology, luckily, is so complex that even if a couple of papers published, on a similar subject look as if they might be the same, there are always nuances that are different because there's such a richness and complexity in biology. So I really think that openness in science, all the way to open access publishing and everything that we do, it benefits enormously by being open.
Bob?
I like to babble and I like to talk about science, and so I mostly do it. Unfortunately, in every field, there's a few people who are not trustworthy, and so those you're a little more reserved with. But when you have your friends and you develop your group of colleagues who you trust and you work with, and so that's really what makes it all worthwhile, is having that group that you can trust and talk and be totally open with.
Thank you all very much indeed. Okay, so there's just a few minutes left, so I just encourage the audience, please do ask your questions using the dialogue box, just before we go into the Q&A. But you've hinted that things might be a little different nowadays, partly the funding environment, making it harder to play around, perhaps. But what do you think? Are there fundamental differences between when you were growing up as a scientist and now? Or is it basically the same? Liz, what do you think?
Well, it's hard to put yourself in the shoes of somebody who's growing up in science now. Although when I look at the experiences of the trainees and the students around me at the University of California, San Francisco, I try to envisage their world, and I think at some level it's really the same. I mean, people are just doing research because they find it extremely interesting, and Even though there is this sort of push towards appliedness. The lesson I had once was when I had an MD PhD student, and so I gave him the project that I thought would be dearest to his MD part of his heart, and he immediately chose a project in my lab which was the most fundamental kind of basic science one. And so I thought, I'm never going to pigeonhole people who are, I would have thought would be wanting to do applied things into that kind of thing. I think that I see the motivations of students as they just want to know. Some people think more in terms of their careers because they do think more strategically than perhaps we did when I was training. But the fundamental, just loving to talk about science or do science, make discoveries, that hasn't really changed. The outside environment, sure, has changed. Digitization, access, the way you can approach things with different technologies, absolutely. Those have made terrific and very exciting changes.
And indeed, we'll be having a whole panel discussion around the idea of how technology and digitization is changing things. But thank you. Andy.
I mean, maybe I would beg to differ a little bit, or maybe accentuate a different point than what Liz just said. I think things have changed, and it's really come into focus, not necessarily just in the last year, but in the last year, in that science is such an integral part of the human race managing to sustain itself. It was always true. I mean, we needed medicine, we needed antibiotics. But I think that the turnaround time is much more so at this point, that we really need we're going to need biotech to generate food. We need to have ways of tracking infections. We need to have ways of dealing with infections and preventing them. We need to have ways of dealing with the environment. A lot of what we're dealing with is what humankind has done, but also a lot of what we're dealing with is that change is really fundamental, is going to happen. And at the present, the way of having several billion people and dealing with that is science. And so it's a mixture of the great need that society has and the great fun and joy that science is. And that's a different equation. And that's where, biotech and academic and traditional industry and government are all areas where science is going to be critical. And I don't think we've actually begun to understand the degree to which we'll need people that are scientifically trained in all those areas. And so I think it's a little bit of a different world. Not that science wasn't important before, but I think the immediacy of scientific discovery being needed on a short term to deal with sustainability issues is something that we're going to be understanding in the next few decades.
Thank you very much. And indeed, we'll be having a panel discussion that will touch on that as the closing panel discussion of all this on the value of science. Thank you. Thank you. Okay, Bob, last word to you.
My early days has changed. So, I grew up right after Sputnik, whenever money was flowing into science, and, everything was fundamental. As we've moved along, I think there's much more emphasis now on having things that at least tie somehow to practicality. For example, the National Science Foundation, now you have to write a statement on broader impacts. So it's, how is this going to influence the field, and how is it going to influence the future of things? So I think part of the thing, as you said, we need more science, and I think more and more there's going to be more and more pressures to go into things that are fundamental, but have an outcome that could really have an effect on the society.
Thank you very much. Sorry, Andy, did you want to come in?
Yeah.
No, I agree with all things, and I fundamental science is really core to a lot of what we're doing. But there's also I think there'll be a shift in a responsibility that scientists have to build their own structures to make the science work for society.
Thank you. So now it's time to move to the Q&A, and this will be moderated by Lee Warren from AISD. And so after a momentary break, the session will be devoted to your questions.
Good morning and welcome to our honored guests, students, and faculty. I am Lee Warren, an instructional specialist in science for AISD. This morning I will be hosting the Q&A session with our distinguished guests, Dr. Blackburn, Dr. Fire, and Dr. Grubes. Good morning, everyone.
Morning.
And with everybody here, I guess we're ready to start with our questions. The first question is from Elena, and she asked, how hard was the journey to where you are today? What were the most difficult parts, and how did you overcome them? We can
Lee, you
start with
probably have to
whomever.
call on us one by one.
Oh, okay. Well, I like this. The teacher in me will come out. Well, I will start with you, Dr. Blackburn.
Oh, I see. That's the problem. I put my hand up, right? Um, I,
Exactly.
how
Exactly.
how hard how hard was the journey? It had hard parts, but the part that was easy was that I did like science and like the idea of becoming a scientist. I didn't really know what that meant, so I had to keep learning all the way through, and I think um, so it's hard work, but when it's hard work that you like doing, it doesn't feel like work nearly so much. The thing that I think I've found so interesting about keeping doing science is that you keep on learning. And so dealing with the difficult parts means just being willing to keep on learning. So, I'm in about grade 65 right now because I'm still learning. And that's what makes it both hard but also a joy to be in science.
Ah, thank you. And Dr. Fire.
I would echo that. I mean, there are parts that are challenging. Sometimes when things don't go right, sometimes when you're in the middle of conflicts of various sorts, that could be a challenging thing. Um, but the science often carries the day in the sense that if things aren't going well, then it pays to go to, in the old days, the library, maybe now the internet, and read some cool science that you might not have known about, to talk to people and learn things that they're doing. Those are good breaks from the hard work.
Ah, thank you. And Dr. Grub.
Yeah, for me, it's I came from a background where most people I knew worked really hard. They didn't have very much success. It was, it was a pretty brutal life, and so relative to that, I've had a phenomenally easy life. And in fact, I went back to my 50th high school reunion, and everybody was shocked I hadn't retired yet, and said you got to work before you retire. So for me, it's been it's been hard. There's been lots of really tough situations, but overall, it's been a journey which has been rewarding, and relative to everything else, it's been really easy.
Ah, and thank you. Sacha asked a question of Dr. Blackburn, and it's kind of a follow-up. Did you face any particular hardships by being a woman in the science field? Um, and how did you overcome those, or have you overcome those hardships?
Well, I think at the very beginning, I when I was, in school, high school, and even before that, and people didn't really expect girls to be that interested in science. And somebody once even said to me, and I'm sorry to tell you, it was a teacher, although not one at my school. They said, "What's a nice girl like you doing going into science?" And the way I coped with this was I kinda got mad. And, but I didn't know how to, express that to the person, so I just kept very quiet. But I was kinda mad inside. It made me very, very determined because I thought that was just a question that made no sense to me. Then earlier in the career, it was much easier because there were more women in, the classes than uh, by then. And later on, it became harder when I think, like in so many careers, you get into much more kind of power-related things, and so it became more challenging, as it does in many professions, as you get more and more kind of senior and established in them. And so there were real challenges. And again, I had to do a lot of learning, and I still have to do a lot of learning.
Thank you, and good luck with that. Um, And from Cassandra Cifuentes, she asked, once again to all of you, so I will be calling on you. Why did you want to do your study in the first place? What prompted you and led you to your work, your research, and to a field in science? This time we'll start with Dr. Grubes.
Yeah, I was always sort of interested in science. I had a phenomenal junior high teacher who still stands out as a person who had a big effect. And so that started things. And then eventually in college okay. And so then in college I discovered organic chemistry. And as I pointed out earlier, it's the one topic I found I was good at. And so it just became a topic to really move forward on, and then finding interesting problems and following them up, and some of them worked out pretty well.
Very good. Dr. Fire.
I mean, one of the wonderful things about being at a university or a college is that you can explore different things you might be interested in. And so I explored as an undergraduate different topics, mostly around some kind of research, but not just science. And I did settle on molecular biology as something I really enjoyed doing and learning about. And so that was a great opportunity to do that. And if I'd taken different classes or explored other things, I might still have enjoyed it, but it might have been a different world for me. So I do look back fondly on that time.
And when you say explore, were you exploring within the College of Science, or were you exploring beyond that field of study?
Well, I mean, I was fortunate to be at a university which Liz was teaching at by the time I left. That you could take any course at any time, pretty much. And so I took things that were in sciences and in other fields too, mathematics, but also history, economics, and I enjoyed all of them. But I certainly enjoyed the molecular biology courses, and it seemed like a field that would be exciting to be a part of. That was the other thing I was looking at the time, whether I enjoyed it and whether I saw myself kind of being a part of it and doing it day to day. And it seemed like something I could enjoy day to day, and that's sort of how I moved into it as a profession, starting with as a college student, just exploring.
And you've done quite well. And and Dr. Blackburn, same to you.
Well, I knew I loved biology. I just loved living things and animals, and then got fascinated by the idea that it's all made up of molecules, and if you understood the molecules, I thought maybe you'd start to understand how life works. So I was kind of excited by that idea and started actually studying biochemistry when I was an undergraduate in college, because I didn't really know that there was a word called molecular biology for a while, and then learned that there was one, and so went on to study a molecule that we all hear about now called DNA, but nobody knew how to sequence the code in DNA back when I was working. And so I started working on very early um, ways of, in a very primitive way, getting what's called the sequence of the DNA, how the building blocks of the DNA are built into a code that specifies, living things. And so just that technology beginning got me very intrigued into, well, what could I use that little primitive technology for, which was to look at the ends of chromosomes, which protect the genes on the chromosomes. We all carry our genetic material as chromosomes, long molecules of DNA. And I just got so intrigued by this subject of how the ends are protected, because it kept on giving surprises and still does. We still don't understand a lot of it. So, and lots of people work on a subject. So the subject just kept drawing me into being curious about how on earth it all was working.
So you ended up down a rabbit hole almost, just spiraling and spiraling. That sounds
Well, actually, the other way
amazing.
around. I sort of took it out into the outer world. First of all, you had to go down the rabbit hole, and then you had to come out and look around the rabbit hole. And so that's where the research has gone. I'm now looking outside the rabbit hole. Rabbit holes are good places to be in science.
They are. They are. We have another question from Maria Noonan, and she asks, "What inspired you to continue your research when your experiments or methods failed or did not produce the results you expected?" We'll go with Dr. Farr. It's your turn to go first.
I mean, so one of the things is that sometimes when things don't go the way you want, there are two things. You try and figure out where to leave things. Maybe you keep doing experiments that you did because you think you didn't do it right the first time. Maybe you redesign those experiments. Maybe you ask a different question and you put the original question, in your pocket somewhere on a list of things in your mind. I want to come back to that. So the work that Greg Mello and I won a prize for, but many people were involved in. I was involved early on in looking at some of the processes that we eventually started to characterize and couldn't make head nor tail out of the data. Tried to publish it, got it rejected initially. Sat in a drawer for a couple of years, eventually put it out to publication. Don't do that if you're a scientist. Always publish it if somebody might be interested. But then it took a few years before other people made advances that let us begin looking at it again. And so there are cases like that where you have to put something aside for a while. There are also cases where just the right approach that day might be good. And so those are what you learn to decide. And the best thing is you don't have to do it all yourself. Early on, there are advisors. Later on, there are colleagues. Sometimes those are very similar. Sometimes there are students, and it's all very similar there too, that you can come to and say, well, what do we do next? And the fact that it's a group endeavor at times is really valuable, and you're not in it alone.
Thank you. Dr. Grubes, and for you.
Yeah. Well, when an approach starts to fail, you can just realize that it's not a very productive one and move on to another one. There's always plenty of great ideas to work on. But the other one is that in many cases it tells you don't understand something, and so then that leads you to go look and try to understand why it went wrong, and that usually leads to a new approach and a new pathway. So it provides you a nice way around it.
And
or it tells you
also,
that
the other
the
thing is,
vacuum manifold is broken.
oh, I'm sorry.
yeah, right. Well, I mean, the other thing is you just have to be an optimist to be a scientist. You have to assume it's going to work and blast on.
Good words. I like that. I have to remember it. And Dr. Blackburn, with you and your research.
Yeah, I think what's been said is absolutely right on and spot on. And I think that the other thing that to think about is that sometimes the technology is just not ready yet for the technical difficulty of an experiment. I'll tell you one thing. I was trying to find proteins that might bind and protect the ends of chromosomes. And they're kind of rare in cells. There's not a lot. So you had to start off with huge amounts of cells and grow them in very large volumes just to get enough to detect these rare proteins. And I tried and tried, and I bought the biggest Waring blender from restaurant-sized Waring blenders to open up these cells to try and get these proteins out, and just couldn't do it. And I had to stop. And the Waring blender broke. That was kind of telling me that maybe, that I was up against a technical wall that later others solved very, very cleverly using very clever genetic tricks to lead them to the right proteins. So that's another reason for failure. You can fail, and it's not because you did anything wrong, or it's just that you might have to actually invent a new technique to solve the problem that you're trying to solve. So it can spur you to really think of new ways of failure can make you think, okay, I've got to think of a new approach, or it's just not going to happen yet. Luckily, biology is filled with questions. There's no shortage of questions to ask.
And it seems that, and what I've heard all of you reference, is that science is a field where you depend on others, other research, other people, others' opinions and curiosities. Would that be a fair statement?
You
It's
both
such a
depend and you're
social thing.
supported by others, and part of your job is to support other people. So it's sort of three aspects to that, all of which are, I think, all of which are good. I think there's a song, "People who need people are the, happiest people in the world." So it's true. Yeah.
And you need time to think yourself, but then, having the brain power of other people to bounce ideas off is incredibly stimulating and really makes the science better. And it's a funny thing, but in some ways, you kind of want competitors because they are advancing the science too. And and so it's a really interesting kind of a mixture of you want collaborators because they will know things that you sometimes can't do, or you just need more hands on deck. It's a multi-person job. And then people who compete in science, and we hear a lot about competition in science, but in a way, we kind of make each other better, by keeping each other up to the mark and competing. Although it can get out of hand, I have to say. And I've always found that the collaborative approach pays off. Um, you know, even though sometimes people don't want to share results, but really collaboration in the long run, everybody ends up doing better by it.
Thank you all. We have another question from Gina Buen. Oh, it just jumped down. Buentipo. And again, it's addressed to everyone. A lot of times we hear that modern problems require modern solutions. Is there ever a time that you think it is important to look to the foundation of science to help in our understanding of studies? Let y'all process that. Ah, I like that. Everybody's smiling. So the biggest,
Absolutely. Absolutely. I
Dr.
mean,
Paul.
there are many times when you work back to a paper that was published 30 years ago, or you work back to something that somebody told you in graduate school who was an expert on the molecule you work on, that was a paper that was published when they were in graduate school. There's a lot of tying back to the past. At the same time, some of the work that was done, you don't have to read, if you're a scientist, you don't have to read every paper that was published 50 years ago, because some of it's summarized, and some of them were methodologies and lessons that were learned and incorporated later on. And so that's a benefit, is not to have to read all hundreds of thousands of papers out there in literature and know everything that everybody knows. So there is a benefit to that. There's also an oral tradition as well as the written tradition of some bits of information and concepts and facts that are passed on essentially from generation to generation, maybe partly written down, but some of it is just what you tell your students and what they tell their students. Um,
Thank you. Dr. Grubbs?
Yeah, you find as you get older, you get to be the person who remembers all the old stuff and takes people back to it. So yeah, so I find myself doing that fairly regularly, of basically recalling something from 30, 35 years ago and saying, let's think about this. So yeah, so there are some of those things. In my field, it's not so much, but occasionally it really pays off.
And Dr. Blackburn?
Yeah, some of the work people did before we had a lot of the modern kinds of technologies that we can use now in experiments and analyzing and digital technologies. People did some very, very good thinking about problems but couldn't really solve them. And so they're really worthwhile looking at. On the other hand, and I think as new technologies come along, you can ask questions in ways that just couldn't be asked before. And some of those old ideas won't necessarily hold up, although a lot of them do, but one of the things about science is it's okay to say, well, what used to be a dogma isn't really a dogma. There was a dogma that says DNA is the information goes from DNA to RNA, which then the information goes to protein. And that is true for great majority of cases. But I worked and co-discovered an enzyme that actually breaks that rule, and it copies RNA into DNA. So, and so do certain viruses. But that was thought to be, well, that was viruses. They're bad anyway and strange. But to have our own cells do that, it doesn't copy a lot of RNA, but it does copy RNA into DNA. It makes real DNA at the ends of chromosomes. So, you have to know the rules really well so that then you understand when something else is going on.
And when you might break them through science and research.
You bet. Yeah, yeah. I mean, science advances by breaking some rules. Some rules, not all. Some have held up very well.
And a question we have from Elizabeth is, what advice would you have for young people starting their journey into the world of science research? We left off with Dr. Blackburn. We'll start with Dr. Blackburn.
Well, two things. It has to be something that you like. You like science, you like doing science, something about it, and not just something that somebody told you is a good idea. So let's assume that's the case. And then it takes a lot of persistence and resilience. We talked about failure, and a lot of times you fail in science because it is challenging and difficult. And so if you can, we talked about earlier how important other people are. So you do want people who support you. They don't have to be necessarily another scientist, but somebody who supports what you think is important, so you can persist and be resilient. So find ways of being persistent and being resilient, because you'll need to do that. But also make sure that this something about science that you do like, you yourself really resonate to.
Thank you. Dr. Grubes.
Yeah, thanks. Yeah, so I think, part of it for someone getting started in science is early on is to understand what you're good at. Find out what fields you're good at. And usually you're going to be happier if you're working in a field you're good at rather than one you're not, which uses a set of skills. And so there's a set of skills that are there that can be used in many different areas. And then, if you can think about then how to find the fields that will use those skills, and then try to then explore from there, and hopefully you'll find a field that gets you really excited, and hopefully you'll find a mentor that can really help you sort of in the early stages of your journey. And I think there are lots of people now. People love to have people follow them in science, and so I think once you've made the commitment, I think you can then find people who can help you on the start of your journey.
Thank you. That mentor and support seems to be big. Dr. Fire.
Yeah, I mean, I would echo that, and also just add to that one looks for both the mentor person who runs a research organization, also the people within the organization. If they're supportive, and if you can also be a member of that group and be supportive, then I think that looks like a very good opportunity. If it seems like it's harder to be a part of the group, then it might not be the best opportunity, and you might want to look for other things. Sometimes it takes going through a bunch of different possible scientific environments to find one that you're comfortable with, and you can optimally learn and contribute to. So
Thank you. Thank you. We're getting close to the end of our time with you. And there's a question that's been asked several times on the board. I think Bex, one of our members, put it best. Was it a shock to get the Nobel Prize? What does it feel like? When did you know that you were even nominated for it? Dr. Grubs.
Yeah, it's one of those things where there's always sciences are great gossipers, and so in the background there's this gossip of who's going to win the prize every year, et cetera, et cetera, et cetera. You know, and unfortunately, there are people who have been hearing that for 40 years, uh, and it can make life pretty tough. So you have to be able to sort of do that, and so you hear that in the background, and so once it happens, it's sort of relief. uh, there's excitement, but there's sort of a relief that that this all happened. And yeah, and it's usually a pretty unexpected.
Well, congratulations.
Yeah.
Dr. Fire.
Well, I mean, it's a very wonderful thing to be recognized by one's colleagues, but there are lots of different recognition things, and there's a joy in having an experiment work, maybe even getting a paper accepted finally after years of going back and forth with a journal and trying to fix all the problems. There's a joy in, if you're a teacher, having a student finish a project. If you're a student, having a mentor, sort of understand what you've done. So I think those are all joys. Prizes are, always a nice thing, but I don't think they're it would be hard to do science just for the prizes. I think one does it for
Oh, yeah.
one does it for a lot of other positive feedback, and some of those are just day-to-day things. Just an experiment that's a pedestrian experiment that you do every day, but when it works, there's a certain joy to that. And seeing bands on a slab of gel that are just straight bands, that's always a joy to me, and that ought to be a
And Dr. Blackburn.
Yeah, I remember I would come into work every day, really excited, thinking, I wonder what the experiment that I had, done the day before, and the results would start showing up experimentally. That I would think, I wonder what's going to happen today. So to have that kind of job is wonderful. And people say things like, well, did you start science to get a Nobel Prize? And I say, you would have to be a really bad mathematician to do that, because think of the number of people who are scientists and the number of people who are Nobel laureates. No way. So I'd heard some rumor that I had been nominated some years before I was awarded this, along with my two collaborative researchers, which was wonderful because we could all have a great party, the three of us. But the thing is that it was I just thought, well, what are the chances? They're pretty low, and so I could torment myself, or I could just kind of push it away and just say, look, the probabilities are low. A lot of people get nominated, so I just kind of pushed that away. So I was pretty shocked when I was called very early in the morning, because it was Swedish time, and in California that means, in my case, it was like 1 a.m. So this was not
Oh my
something I
goodness.
really was thinking about. Or in fact, I thought it was because my mother-in-law, who just had her 95th birthday, I thought maybe something had just happened because we'd had a big party that day. So I was pretty relieved that's what it was, and not bad news about my elderly mother-in-law, who lived 6 more years, by the way.
Yay. Well, thank you all. It is such an accomplishment, and we thank you for taking the time to be with us today. It has been a pleasure. You have enlightened us, and paved the way. So thank you again. And with that, I believe our session is over.